Assalamu alaikum. Today in this video I'm going to tell you the process of bilirubin formation, how bilirubin is formed in the body from red blood cells. It is a very important topic with regards to the paper of biochemistry because it is repeated constantly in the past papers. Let us see how it starts. We know we have red blood cells in our body, 4.5 to 5 millions and a lot of number. They have a lifespan of 120 days. After completing their lifespan of 120 days, which RBCs have, they break down into their components. RBCs are made up of basically hemoglobin, which contains an atom of iron in the center. So when they break up, they release hemoglobin, which consists of heme, globin, and along with the atom of iron which was present in the center of the hemoglobin. These three are the components. After breakdown of the RBCs, it occurs in the reticuloendothelial system of our body, which is explained in the a different video, not here. Now let us see what happens with all these three things. Iron is stored in the body. If it is to be utilized, then it is utilized in the body. It is stored in the form of ferritin and it is utilized along with the transferrin, so it is a different case. Globin, which is another product of the RBC degradation, it is also an amino acid. It is stored in the body for future use of the RBCs, whereas the left one is the heme. How is the heme then converted? It is then processed in the body and converted into biliverdin. This all is happening in the blood. Heme is converted to biliverdin. How this process occurs? First of all, atom of iron is removed, which I have shown here. Carbon monoxide molecule is removed. The coenzyme here is nicotinamide adenine dinucleotide phosphate dehydrogenase. It is converted into NADP, and heme oxygenase is the enzyme here. After all these reactions, which mainly the two things which you have to remember definitely is the enzyme heme oxygenase and the coenzyme NADPH. Nothing else is needed. Heme is converted into biliverdin. After biliverdin formation, remember it is very important MCQs of biochemistry that what is the first pigment of RBC degradation? It is not bilirubin, it is biliverdin. It is a green colored pigment which is first produced in the process of RBC degradation. After biliverdin formation, it is readily converted into yellow colored pigment bilirubin in our body. It requires the enzyme biliverdin reductase. First of all, I showed you here that enzyme was heme oxygenase, but from biliverdin to bilirubin, enzyme is the biliverdin reductase. Now we have reached to the bilirubin stage. It is an insoluble thing which is not soluble in water and it is lipid soluble, so it cannot be transported without any aid in the blood because blood is plasma and plasma is water. So what do we need? We know that in plasma of the blood we have different plasma proteins, albumin, globulin and so on. One of which is albumin, which is a very good transporter. This bilirubin combines with the albumin, which I have shown here, bilirubin combination with the albumin, and then it sits on the surface of albumin and is transported to the liver. This all is happening in blood. I have already told you RBC degradation. Two components are stored. Heme is converted into biliverdin with heme oxygenase enzyme, and biliverdin is the first bile pigment. It is then converted into bilirubin by biliverdin reductase. This is then bilirubin then combines with the albumin, which is a plasma protein, and it goes into the liver. In liver, this bilirubin detaches with the albumin. Bilirubin then attaches with another thing which is present in the liver, that is glucuronic acid. This bilirubin combines with glucuronic acid to form bilirubin diglucuronide. It is another product which is formed during the RBC degradation, bilirubin diglucuronide. It occurs in the liver in the presence of enzyme UDP glucuronyl transferase. UDP glucuronyl transferase is the third enzyme. First was the heme oxygenase, second was the biliverdin reductase, and third is the UDP glucuronyl transferase, which forms the bilirubin diglucuronide. Now this bilirubin is very easy to travel. First of all, individually it was lipid soluble molecule and not was able to travel in the plasma, but now bilirubin diglucuronide is a water soluble thing. It can easily travel in plasma without this aid of albumin. Now albumin is kicked off from the whole story. Bilirubin diglucuronide then goes into the bile, which is present here. Through common bile duct, it is released into small intestine of our body. Here is the small intestine. When bilirubin diglucuronide comes here, it is again released, broken down into its components, which is bilirubin and glucuronic acid. Now this bilirubin is converted by the action of some bacteria which are present in the small intestine into urobilinogen. Urobilinogen is another product during this RBC degradation. Okay, the first here I showed you bilirubin diglycuronide. Bilirubin diglycuronide in small intestine is again broken down into bilirubin and glucuronic acid, and this bilirubin is converted into urobilinogen. This urobilinogen is then converted, part of it, not all of it, is converted into stercobilinogen. Stercobilinogen goes to the feces and converts to stercobilin. The characteristic brownish yellowish colored of stool or the feces is because of this component stercobilin. During jaundice, this component is not present, especially in the post-hepatic jaundice, because due to which we have white chalky colored stools, not the characteristic color which is brown. I said part of it was converted into stercobilinogen. Remaining of the urobilinogen is reabsorbed into the body in our portal system, which is that it goes into the liver again. From here, it goes to the portal system to kidneys. From kidneys, it is converted into urobilin and is excreted in the urine. Urobilin is that component which gives characteristic yellow color to our urine. In the absence of this component, urine color is lost, and in absence of stercobilin, the color of stool is lost. In this way, whole of the bilirubin is excreted in the form of, from the body in form of stercobilin and urobilin. A very important MCQ again, that what are the excretory forms of bilirubin? These are stercobilin and urobilin. Never bilirubin is excreted in its own form. So whole of this story occurs. First bile pigment was biliverdin. Second is the bilirubin. Then I said the third thing which was important was bilirubin diglycuronide. Then it was broken, and fourth thing was urobilinogen. After that, stercobilinogen and urobilin and stercobilin. Enzymes were heme oxygenase, biliverdin reductase, UDP glucuronide transferase, and bacterial enzymes in small intestine. I hope the story is quite easy. If you have any queries, you can ask. Thank you so much.